Hello, this is Mahendra Didia. In this portion of the book on liquid formulation, we continue to cover emulsion formulation using integrated approach to formulation development. This is the last module in the book, module 16. It focuses on quality control and clinical study formulations. In this particular submodule, module 16.2, we focus on clinical study formulations. We'll look at both pharmaceutical formulations as well as cosmetic formulations. Pharmaceutical formulations utilize in vitro release testing prior to selection of the formula for clinical studies. This method is recognized by FDA and other authorities. We will review several of these methods. In vitro release test, IVRT methods, are generally correlated to clinical studies, specifically pharmacokinetics or bioavailability clinical studies. FDA recommends IVRT as part of new drug application. USP apparatus 2 paddle method has a sample holder at the bottom and a membrane is placed in the holder. The product is filled in the bottom portion of the container and reservoir containing medium is filled and stirred. This is a suspension cup method. Is dialysis membrane method? A dialysis tube is used in which the drug is filled. The leaching of the drug from dialysis tube into the medium is studied. And apparatus 4 flow-through cell is used. Vertical diffusion cell, France diffusion cell is very commonly used. In this case, donor chamber that contains the drug is separated by a membrane and there is a receptor compartment in which the media is filled and the samples are taken at given time intervals. Let's look at dialysis method. In direct dialysis method, drug is placed into the dialysis tubing and it is immersed into a receptor media. This can be centrifuged and drug will be released and free drug is assayed. Reverse dialysis method, drug infuses into the dialysis tube. The concentration of free drug is measured. Reverse dialysis method in which drug is in the solution and that leads into the dialysis tubing. There's an apparatus. It is a flow-through cell in which the media continuously flows through and is recirculated. The reader is requested to review module 13.4 on in vitro methods. Let's look at suspension cup method using apparatus 2, and then we'll look at France diffusion cell in this particular portion of the slides. What you will see is a cup filled with cream, and then membrane is put on top of it, cascade is put in, and it's sealed, and then cup is lowered into USB apparatus 2 for measuring release of the drug. So let's look at the video. The membrane is being put on to the cup that contains cream. Gasket is placed to hold the membrane tight, and then it's sealed properly. The cup is inserted into a special apparatus tube, and one can see the stirring mechanism here. The media is filled and stirring mechanism is started, and then samples are taken at time intervals to measure the drug release from the cup. Let's look at France diffusion cell. The donor compartment, drug is placed on top here. A membrane is placed and is tightly held. Here is a receptor compartment and it is stirred. Samples are withdrawn at given time interval as drug is released. Expanded picture of the fast diffusion cell. Drug semi-solid metrics such as emulsion, suspensions, membrane. France diffusion cell systems are automated, and let's look at the video of that. Multiple cells are inserted into the system that's held at constant temperature with the proper systems are calibrated and data is generated by automated analysis. Let's look at IVRT method dialysis, sac, and flow-through cell. And here we'll show the reverse dialysis of iodine from a solution into the dialysis tubing. In pharmaceutical, these are calibrated system. Dialysis membrane is placed in a vertical position and media flows through in a closed loop system. So let's look at dialysis experiment first to understand what is reverse dialysis. And it will transfer into the dialysis tube containing starch. Iodine reacts with starch, and one can visually see that. So iodine is added, dialysis tube is placed, and after several minutes, we will see, in this case, iodine selectively going into the tubing. This is reverse dialysis. So let's look at flow-through cells that are commonly used in pharmaceutical systems. So there are cells here. A glass bead is added and sealed. Entire system is then placed into a water bath at a controlled temperature. In closed loop system, media is circulated through the filters into the system, and data is analyzed by graphing it. Analysis of drug is conducted, and cumulative percent or differential percent is measured. In vitro release evaluation of reference and a test drug formulations. In order to compare that, FDA requires that complete and comparable release reach the plateau and the graphs be evaluated using statistical method such as F2 calculation. Let's look at IVRT profiles of risperidone parenteral nano formulation. Reader is requested to review module 13.2 for additional information on the drug and the physical attributes of the formulations. In this case, two different formulations are being looked at: lecithin, polysorbate as surfactant or emulsifiers, and only lecithin. Reverse dialysis flow-through cell USB apparatus was used. In this case, drug is in the solution and drug then transports into the dialysis tubing and the concentration of drug is measured. Percent drug released is shown here over 3 hours. One can see that risperidone formulation with lecithin and polysorbate 80 is shown as a blue curve, and it releases fairly fast and continues to increase over the time frame. As compared to only lecithin used as an emulsifier, drug release is slow initially, and after about an hour, both systems appears to release drug at about the same time though there's some slight variation shown here. This type of information is then used to select the appropriate formulation for clinical study. Let's look at IVRT and in vivo blood level data using preclinical model for carbamazepine nanoemulsions compared to emulsions. Preparation and characterization of nanoemulsion. Emulsion is prepared by dissolving carbamazepine in oil. Oil is added to the water phase. This is a coarse or microemulsion. It is then homogenized at ultra high pressure to produce nanoemulsions. Physicochemical characterization includes particle size, zeta potential, viscosity, and separation through centrifugation using creaming index. IVRT physicochemical mechanisms through dialysis tube is shown in this portion of the slide. The drug is in oil phase. This is the interface, and external phase is an aqueous phase. When drug is exposed to dialysis tubing or biological membrane, drug migrates to the interface, and if drug aggregates into the aqueous phase, and then it transports through membrane into the receptor compartment. Dialysis membrane is used, drug is placed in the membrane sac, and drug release is measured. In vivo release is conducted in rabbit. Rabbit are injected with the emulsion, nanoemulsion, as well as the drug is assayed in the plasma. Let's look at the results. Let's look at IVRT dialysis method. The nanoemulsion is shown is in red square, and it shows rapid release initially and continues to release and reaches plateau around 12 hours. The microemulsion shows slower release and reaches plateau. Carbamazepine serum concentration in rabbit are shown here. Nanoemulsion and emulsions. These curves look different. Nanoemulsion is released rapidly and maximum concentration is reached in less than an hour, followed by slow release of residual drug. Emulsion, maximum concentration of the drug is achieved at about 12 hours. It starts with slow release of the drug. Let's look at cosmetic formulation. In this case, 3 different formulations are compared to Canoderm, a commercial product. The products are pickering cream, surfactant base, and surfactant base cream. Pickering cream uses solid particles separating oil and aqueous phase. Surfactant-based replica of Pickering cream, SCC, replaces solid particle stabilizers by emulsifiers. Surfactant-based cream has a different composition, contains same emulsifiers as SCC. Oil components are replaced by dimethicone, a siloxane. All these products show shear thinning behavior. Canoderm has a higher viscosity as compared to SC3, which contains dimethicone. Hydration of exercise skin shows that Canoderm has the highest percent water as compared to that SC3 shows smaller water. Sensory perception evaluation in human was conducted as shown here. Several attributes were measured as described by human volunteers, from moisture, spreadability, thickness, absorption, stickiness, residual coating, greasiness, slipperiness, and so forth. Both SC3 and modified Pickering cream showed more acceptance for first 2, moistness and spreadability, whereas slipperiness and softness was less acceptable. Pickering cream was less acceptable in several criteria as shown here. Reader is requested to refer to module 13.2 for rheology and sensory characterization also. So this data then can be used to further optimize formulation shows more acceptance in all tests that are conducted.